right, and I'm gonna let them in. I'll monitor microphones and things like that, and then you, Jeff, can just take it over whenever you're ready.
Okay, thank you.
All right, here we go.
Hello and welcome to today's workshop. We'll be starting the workshop here in a few minutes. I want to give time for more attendees to enter the room. Um, Hello and welcome to today's CFSRE Current Trends in Forensics Toxicology Workshop. I am Jeffrey Williams, the manager of the phytocannabinoid R&D group at Cayman Chemical. Cayman is sponsoring today's workshop, and I'll serve as moderator for today's speakers, Alicyn Stothard and Jonathon Bassman. They will be discussing semi-synthetic cannabinoids and what we know about their metabolism. This includes some in vitro experiments on a series of semi-synthetic cannabinoids we conducted at Cayman Chemical, and we're excited to share those results with you today. If you have questions during today's workshop, I invite you to include them in the group chat at the bottom of the screen, and I will direct them to our speakers at the conclusion of today's workshop. I'd like to introduce our speakers for today. First, Alicyn Stothard graduated from Central Michigan University with a degree in biochemistry, and she recently completed a Master's of Science in Chemistry from Eastern Michigan University. She is an associate scientist here at Cayman, and she's had over 4 years of experience making reference standards for naturally occurring phytocannabinoids, as well as a large number of the semi-synthetic cannabinoids that appear in today's talk. Jonathon Bassman is our second speaker. He has a bachelor's degree in chemistry from the University of Michigan, as well as a master's of science in chemistry from Eastern Michigan University. He has over 14 years of experience at Cayman working on the synthesis of a diverse number of novel psychoactive substances, their metabolites and internal standards. He has recently been working as part of a collaborative effort here at Cayman to conduct in vitro studies of new NPS compounds to identify presumptive metabolites of interest. Alicyn will start today's talk titled Semi-Synthetic Cannabinoids, an emerging NPS overview and case study on in vitro metabolism. Alicyn, please take it away.
Thank you, Jeffrey. To understand the semi-synthetic cannabinoid landscape, it is important for us to define the terms phytocannabinoid and semi-synthetic cannabinoid. Here, I have the structure of delta-9 tetrahydrocannabinol, our primary phytocannabinoid of interest, and delta-8 tetrahydrocannabinol, one of the most common semi-synthetic cannabinoids. Below the structures, we have provided the item numbers for our associated products, here and throughout the presentation. Phytocannabinoids are naturally occurring cannabinoids found in the cannabis plant, whereas semi-synthetic cannabinoids are compounds synthesized from isolated hemp-derived materials. A good example of the formation of a semi-synthetic cannabinoid is the conversion of CBD to delta-8 THC. There is no biosynthetic pathway in the cannabis plant to provide delta-8 THC, which makes it a semi-synthetic cannabinoid. We will define semi-synthetic cannabinoids by their relationship to delta-9 THC. So we will begin by describing the biosynthetic process of forming delta-9 THC. Here, we have a general overview of the cannabis biosynthetic pathway. Geranyl transferase catalyzes the formation of cannabigerolic acid from levotolic acid, and from CBGA, various synthases make cannabichrominic acid, cannabidiolic acid, and tetrahydrocannabinolic acid A. These acids can be decarboxylated by heating. This is the process that provides us with delta-9 THC, the major psychoactive component of the cannabis plant. Semi-synthetic cannabinoids can be separated into three key categories: isomers, homologs, and analogs. Each of these categories will be defined in their relationship to delta-9 THC. Starting with the isomers, some of these compounds, such as delta-8 THC and delta-10 THC, may be more familiar semi-synthetic cannabinoids to many people. Isomerization of delta-9 THC can occur due to ring strain associated with the olefin. Under acidic conditions, this leads to isomerization of delta-9 THC to delta-8 THC, which relieves the ring strain. Other isomers can be formed under various reaction conditions. We will discuss several isomers and their metabolism in upcoming slides. Next, we will discuss the homologs. The homolog compounds retain the core THC structure with variations to the alkyl chain length. For example, tetrahydrocannabivarin, or THCV, has a 3-carbon aliphatic chain, as opposed to the typical 5-carbon aliphatic chain associated with THC. While several of these homologs, such as THCV and THCP, have been identified in plant material, they are present at very low quantities. These homolog compounds cannot be synthesized from CBD, but other synthetic routes have been established. The starting materials for these syntheses have gotten much cheaper over the last several years, making it likely that these homologs are purely synthetic when identified in cannabis products. Lastly, the analogs also retain the core THC structure, with various synthetic modifications to either the phenol or the olefin. The HHC, or hexahydrocannabinol, and acetate compounds are common semi-synthetic analog components of cannabis products such as vapes or gummies. There are also compounds at the intersection of these categories, such as hexahydrocannabiforol, or HHCP, which is both a homolog and an analog, and delta-8 THC acetate, which is both an isomer and an analog. We will continue our discussion of the semi-synthetic cannabinoids with a synthesis and a metabolism case study of 4 THC isomers that are increasingly prevalent in the cannabis market. Before we get into the metabolism case study, we will discuss the formation of the delta-10 isomers of delta-9 THC. Delta-10 THC was first described in the 1970s, and to form Delta-10 THC, Delta-9 is isomerized under basic conditions to provide the 9S and the 9R C9 epimers. These compounds are diastereomers, and as diastereomers, they are separable under reverse phase conditions. These compounds have been reported to have no psychoactivity. Additionally, no metabolism data has been previously described in the literature. Regardless of the lack of psychoactivity, these compounds are marketed in products such as vapes and gummies. The Delta-10 THC diastereomers can be further isomerized under acidic conditions to form the 9S and the 9R delta-6A10A enantiomers. As these are enantiomers and mirror images of each other, these compounds are not separable by normal methods, but require chiral chromatography. Literature has reported the psychoactivity of the 9S enantiomer to be 10 to 30% of the psychoactivity of Delta-9 THC, with minimal to no psychoactivity for the 9R enantiomer. And in this case also, no metabolites have been described. Due to the presence of the delta-6A10A and delta-10 THC compounds in cannabis products, and the fact that many of the products marketed as delta-10 likely do not differentiate between delta-10 and delta-6A10A, there is significant value in describing their metabolism. Here, I will pass the discussion to my colleague, John Bassman, for a discussion of the metabolism background and a following case study.
Thank you, Alicyn. Good afternoon, everybody. So yeah, with the ever-increasing prevalence of semi-synthetic cannabinoids in commercial products such as electronic vaping pens and gummies, there's been an increased interest in their detection, analysis, and insight into their metabolism. And so today I'm going to talk a little bit about the metabolism of a set of semi-synthetic cannabinoids, and in particular, a case study that we performed in our labs here at Cayman Chemical using an in vitro metabolism method with human liver microsomes to gain some insight into the metabolism of delta-10 THC and delta-6A10A THC. Now, before I get into that, I think it would be useful to give a brief overview of some of the reported metabolism that's been done in the past on more traditional cannabis-based components such as delta-9 THC, the main psychoactive component of cannabis, and delta-8 THC, which is a major degradant product. And so the metabolism of delta-9 THC to form phase 1 and phase 2 metabolites has been extensively studied dating back to the early 1970s. Various models have been used, both in vivo and in vitro. And regarding the in vivo studies, the animals as test subjects that have been analyzed were mouse, rat, guinea pig, rabbit, monkey, and eventually human. Organs and tissues that were experimented on were liver, brain, intestine, and lung. Liver, of course, being the main organ responsible for metabolism. In vitro methodologies made use of either liver homogenates, which is essentially fresh liver tissue that's mechanically pulverized and homogenized, or liver microsomes that are the tiny vesicle-like structures containing cytochrome P450s responsible for metabolism. And so some of the earliest work was performed by Wall and his colleagues using in vitro models with either rat liver or rabbit liver homogenate. In one of his early studies, he first was able to detect two major metabolites of delta-9 THC for the first time, 11-hydroxy delta-9 THC and a dihydroxylated metabolite, 8-alpha-11 dihydroxy delta-9 THC. He then used a rabbit liver homogenate model and was able to find 3 metabolites of reasonable significance, the same 2 as the rat liver model in 11-hydroxy delta-9 THC and 8-alpha-11 dihydroxy delta-9 THC, and a third metabolite in 8-beta-hydroxy delta-9 THC. And so I've starred these 2 positions on the delta-9 THC molecule just to indicate where the hydroxylation has occurred. Now, delta-8 THC, as Alicyn mentioned, is not formed or naturally in the plant material. However, it is a major degradant product of delta-9 THC usually formed during storage and handling conditions. And therefore, it finds its way into commercial products. Thus, its metabolism has been extensively studied as well over the years. So various researchers have used both in vivo and in vitro methodologies. Starting with Burstein in 1970, he used a in vivo rabbit liver model. And what they were able to find was that the metabolism of delta-8 THC solely gave a single metabolite product in 11-hydroxy delta-8 THC. Around the same time, Foltz and his colleagues used an in vitro model with rat liver homogenate, and they also only found one major metabolite in 11-hydroxy delta-8 THC. Now, Wall, who did the work on the previous slide on delta-9 THC, employed the same tactics and experimental conditions for delta-8 THC, and he was able to find 3 metabolites, 11-hydroxy delta-8 THC, and 2 dihydroxylated metabolites, 7-alpha and beta, 11-dihydroxy delta-8 THC. And in this case, 11-hydroxy delta-8 THC was the major metabolite. And again, I've starred the positions as above where hydroxylation has occurred. And these are known as the allylic positions, in other words, adjacent to a double bond. And so from a synthetic organic chemistry perspective, it's not surprising that hydroxylation would be occurring at these positions, as allylic positions are known to be, or positions adjacent to functionalities such as double bonds, triple bonds, and aromatic rings are known to be susceptible to chemical reactions like oxidations. That being said, metabolism studies are capable of performing oxidations on parts of molecules otherwise generally considered unreactive, such as straight hydrocarbon pentyl chains. And so that was demonstrated by Burstein in 1972 using an in vivo rabbit model. He administered rabbits with delta-9 and delta-8 THC and analyzed the urine samples over time. And what they discovered was, on top of major metabolites such as 11-hydroxy delta-9 and delta-8 THC, there was also hydroxylation occurring on the pentyl chain. And most of these studies were concerning phase 1 metabolism, meaning simple hydroxylation or di or tri-hydroxylation. However, which would produce, in these cases, the primary metabolites 11-hydroxy delta-9 and delta-8 THC. However, there can be further oxidation reactions occurring in a more downstream way to produce phase 2 metabolites, forming carboxylated materials such as 11-carboxy delta-9 THC and 11-carboxy delta-8 THC. And these have been reported to be the major phase 2 metabolites of these components. And those are generally detected either as the free carboxylic acid here or as the glucuronides, which occur when glucuronic acid interacts with the free carboxylic acid of the substrate, forming a much more water-soluble material that can be processed by the kidneys and excreted. And so now that we have a reasonable idea of the metabolism of the more traditional and well-known substituents of cannabis, such as delta-9 THC, there's been a lot of intrigue into the analysis of the metabolism of semi-synthetic cannabinoids, such as delta-10 THC and delta-6A10A THC. Now, there's actually little to no reported literature or studies on the metabolism of these isomers, whether in vivo or in vitro. And so what we wanted to do here at Cayman was perform a case study internally to try to gain some insight into what the metabolism of these materials would provide. And based on the past metabolism of delta-9 THC and delta-8 THC, along with some other reported metabolism of semi-synthetic cannabinoids like HHC that Alicyn will get into later, we hypothesized that the major metabolites would be hydroxylated at the 11 position, or on the methyl group attached to the cyclohexene ring. We also wanted to gain insight into some of the other metabolite products that would be occurring. So the study goal was to identify the probable phase 1 metabolites of delta-10 THC and delta-6A10A THC. However, before getting into that study, we first want to do an initial test case on delta-9 THC to see if our experimental conditions would give results that are in line with some of the past work that's been done since the 1970s, as I discussed. And so our experimental setup involved mixing human liver microsomes with NADPH cofactor, phosphate buffer, and our substrate of choice, in this case delta-9 THC to start, and incubating at 37 degrees C for 1 hour. The reactions were quenched with acetonitrile and centrifuged, and the resulting supernatant was injected into a UPLC system, and the resulting products analyzed by mass spectrometry with an Orbitrap system and selective ion monitoring. So selective ion monitoring is a useful technique with mass spectrometry to isolate specific target masses of products, with the goal to kind of simplify the potentially chaotic mess of analytical results. And so based on the metabolism reported on delta-9 and delta-8 THC, we presume that we'd primarily be seeing monohydroxylated, dihydroxylated, and possibly carboxylated, even though that's a phase 2 metabolism procedure. And those masses to look for would be 331 for monohydroxylation, 345 for carboxylation, and 347 for dihydroxylation. The standards that we were able to use that we had on hand were 11-hydroxy delta-9 THC, 8-alpha and 8-beta hydroxy delta-9 THC, and 11-carboxy delta-9 THC. And what we saw after running the experiment was 3 metabolic products, one of which was by far the major product at 13.52 minutes, as you see in the chromatogram in the central part of the screen. Now, using SIM, we did not observe any appreciable amount of dihydroxy or carboxylated metabolites, and that was the case for all the other substrates like delta-10 and delta-6A10A-THC as well. And so we primarily focused throughout the project on just monohydroxylated species. So now, as we pick each product apart by its mass spectrometry results, we see the major metabolite at 13.52 has a retention time match to the 11-hydroxy delta-9 THC standard. And that mass spectrum of the standard is in the upper right. The mass spectrum of the sample at 13.52 is below that. And you can also see that there's a near identical spectral match. And so this was a great result to start our project, as not only do we get a retention time match and a spectral match to the standard, it ended up being by far the major metabolite, which is what we would hypothesize to be and what was in line with past literature results. Now, looking in more detail at the mass spectrum itself, we see a set of major fragments, the presumed chemical structures of which are displayed on the bottom right. Now, the one highlighted in blue has a mass to charge ratio of 193. And as you can see, there's no additional hydroxyl group on either the aromatic ring or the pentyl chain. So that indicates that hydroxylation is occurring either around the cyclohexene ring system or on the methyl group, which is obviously the case for 11-hydroxy delta-9 THC. Another fragment of note is a large 313 signal, which would indicate loss of the hydroxyl group. And there's a very small 331, which is simply the molecular ion. And if you look at the scheme on the lower left, we can make sense of that by noting that the hydroxyl group is added to the allylic position adjacent to the double bond. So it has a high likelihood of cleaving due to the resultant charge being stabilized by the nearby double bond. Now, if hydroxylation occurs further or more distal to the double bond, such as at the 7 or 6A or another position, it's going to be less likely to cleave, and there will be a larger 331 fragment ion signal versus a 313. Now, looking at the other products, the first one at 12.9 minutes, we have the mass spectrum in the upper right. Again, we see a set of major fragments with the presumed chemical structures on the lower right. Again, I have a fragment highlighted in blue, and that has a mass to charge ratio of 191. And so what that seems to indicate is hydroxylation on the pentyl chain. During the fragmentation process, the hydroxyl group is cleaved and a double bond is formed. producing a 191 fragment ion. Additionally, there tends to be with the 191 a paired 257 ion mass. So usually seeing a 191 and a 257 in the mass spectrum would indicate likely hydroxylation on the pentyl chain. And lastly, the signal at 14.77 minutes has the mass spectrum shown here. Now, we don't have any spectral match to the standards. However, we know that it's certainly not 11 hydroxy delta 9 THC, and it's not 8 alpha or beta hydroxy delta 9 THC. But we do see a 193 ion signal, which would indicate hydroxylation around the cyclohexene ring system as opposed to the chain. There's also a large 331 to 313 ratio, which, as I said, likely indicates hydroxylation further from the double bond, for instance, at the 7 or 6A positions or somewhere else. So now that we verify that our experimental conditions would give reasonably accurate results, at least compared to the past literature, we felt confident using those same conditions to test out the metabolism of semi-synthetic cannabinoids. So starting with 6AR, 9R, delta 10 THC, we see a chromatogram in the central part of the screen that shows 4 different signals. So looking at the major metabolite at 13.86, as I stated earlier, we would anticipate and hypothesize that hydroxylation at the methyl group would be the major metabolic product. And what we see is a retention time match to our standard 6AR, 9R, 11 hydroxy delta 10 THC. And the mass spectrum of our sample is on the lower right, the mass spectrum of the standard 11 hydroxy delta 10 THC on the upper right. So as you can see, we don't see an exact identical match. However, there are similarities, such as the 193 fragment ion signal, indicating hydroxylation either on the methyl group or around the cyclohexene ring system, and a large 331 to 313 ratio. Also is a 301 signal, which seems to be unique to 11 hydroxylated delta 10 and delta 6A10A species. However, as you can see, the mass spectrum is a bit messy, and even in the chromatogram, there does appear to be a bit of overlap. So I think there is a mixture of hydroxylated species in our sample. However, we are confident that there is at least some 11 hydroxy delta 10 THC in that mixture. Now, looking at 2 other of the products in tandem at 13.7 and 14.16 minutes, we have those mass spectra on the right. So they're distinct from each other, but they share commonalities such as a 193 signal, again, indicating likely hydroxylation somewhere around the ring system, and a large 331 to 313 ratio, indicating hydroxylation further from the double bond. So we anticipate that either or one or both of them are hydroxylated at the 8 position or the 7 position, and they can be either in the alpha or beta orientation. However, without standards at this time, we cannot definitively verify that. Lastly, the signal at 13.35 has the spectrum shown to the bottom right. As we see, we have that pairing of 191 and 257 fragment ion signals, which likely indicates hydroxylation along the pentyl chain. Now moving on to 6AR9S delta 10 THC, what we see is a chromatogram with 3 major signals, with a bit of overlap is what it seems like, especially for the first 2 signals. Now the major signal at 14.13, which again we hypothesize would be hydroxylated at the 11 position, has the mass spectrum on the lower right. Now we don't have the standard to be able to do a direct spectral comparison, as we ran into synthetic difficulties in making it. However, that work is still ongoing. But what we do see is a 193 fragment ion signal in the central part of the spectrum, indicating hydroxylation either on the methyl or around the ring, and a large 331-313 ratio, again, indicating likely hydroxylation further from the double bond. Now, the next most abundant signal at 13.57 has the mass spectrum shown below. It also has a 193 signal, which would indicate likely hydroxylation around the cyclohexene ring. However, due to what appears to be some overlap in the chromatogram and a reasonably messy mass spectrum, it appears to be a mixture of multiple hydroxylated species. And then finally, the signal at 13.21, that also looks like it's possibly a mixture of materials based on the broadening in the chromatogram. That has a mass spectrum shown here, and you can also see that it clearly is a mixture of hydroxylated species, as it has both a 191 and a 193 fragment ion signal. And so it's likely a mixture of hydroxylated species on the pentyl chain as well as around the ring system. So now moving on to the 6A, 10A series, we have for both the 9R and the 9S, the chromatograms of each on the upper right. And as you can see, they have near identical chromatographic outputs, with producing two major signals, one at 13.31 minutes and one at 13.7 minutes. And they have the mass spectrum of each individual signal is identical to each other from 9R to 9S. So I've only displayed one of the spectra here below. And so the signal at 13.31 shows a mass spectrum that contains the 191 and 257 pairing, which would likely indicate hydroxylation along the pentyl chain, as I've discussed. And now looking at the major metabolite at 13.7 minutes, which again, we hypothesized to be hydroxylated at the 11 position being the major metabolite. We have that mass spectrum shown below. Now we don't have either reference standard of the 9R or the 9S 11-hydroxy delta 6A 10A THC. However, we do have the mass spectrum we do have the standard of 6AR 9R 11-hydroxy delta 10 THC. And as you can see, these mass spectra are actually quite similar, strikingly similar really. And you can sort of make sense of that by looking at the chemical structures. If we presume this is the 6A 10A is hydroxylated at the 11 position, it would not be surprising to have a mass spectrum closely matching the delta 10 scenario. As you can see, the chemical structures are nearly identical except for the placement of the double bond one carbon position away. And so that wraps up some of the results of our in-house case study that we performed. And so what we can conclude from our results are that the human liver microsome metabolism experiments primarily produce monohydroxylated metabolites. The metabolism of delta 10 and delta 6A 10A THCs appear to give 11 hydroxylated species based on similarities to available standards, but they also, the experiments also form hydroxylated species along the pentyl chain and elsewhere around the cyclohexene ring system. And specific ions to look for identification are 193, which would indicate hydroxylation either on the methyl group or around the cyclohexene ring system, or 191 fragment ion signal, which would indicate hydroxylation along the pentyl chain. Now, future considerations for similar work would be to ideally obtain currently unavailable standards of the presumed metabolites being formed, method development for improved chromatographic resolution, and possible in vivo metabolism studies. And so the work presented here can also be found in written form that we wrote ourselves at Cayman Chemical, and you can either go directly using the QR link on the bottom right to find the monograph, or go onto the Cayman Chemical website, caymanchem.com, and follow the literature tab followed by the monograph tab. And it'll be one of the main monographs, or one of the first monographs that you see. So now, before I hand the microphone off back to Alicyn, I just wanted to note that there was a very recent study published in the Journal of Analytical Toxicology that actually did identify 6AR 9R 11 carboxy delta 10 THC in authentic urine specimens. And that is, of course, the phase 2 metabolite arising from oxidation of 6AR 9R 11 hydroxy delta 10 THC. And they were able to use a standard that we provide to verify their studies. And so that essentially validates that our work is going into the right direction and that these metabolites indeed are being formed. And so that was really interesting. And now I will pass the floor back to Alicyn.
Thank you, John. So we've discussed several isomers and their metabolites. Now we will transition into discussing prevalent analogs, such as the hexahydrocannabinols and the THC acetates and their metabolites. The hexahydrocannabinols, or HHCs, are of specific interest due to their explosion in the cannabis market, both in the US and abroad. The HHCs are often marketed as hemp-derived, as they can be synthesized starting with CBD. THC can be formed through acid-catalyzed cyclization of CBD, and then either delta-8 THC or delta-9 THC can be hydrogenated with a metal catalyst to provide the 9R and the 9S diastereomers. These diastereomers are fully separable by normal phase methods, and activity of these compounds has been well established, as we will see on the following slides. Literature since the 1970s has reported on HHC activity, with early reports describing activity of the 9R diastereomer as comparable to delta-8 THC. This table summarizes activity data from a rhesus monkey study, and as you can see, delta-9 THC requires the lowest dose for psychoactive response, with delta-8 THC and 9R HHC requiring double the dose for the same effect. The 9S HHC, however, requires an almost 20 times higher dose for a similar psychoactive response. This scientific reports paper from 2023 supports these conclusions. As you can see in these results from this mouse tetrad test, the 9R HHC, designated by the black bar, exhibits a greater effect than either the 9S HHC in gray or the vehicle in white. Additionally, there have been studies that support the activity of these HHCs based on the binding to the CB1 and CB2 cannabinoid receptors. The binding of 9R HHC is comparable to that of delta-9 THC for both cannabinoid receptors. As you can see from the graphs, the red and green lines designating delta-9 THC and 9R HHC follow the same path, while the blue line, designating the 9S HHC, is much lower. The Ki values for delta-9 THC and 9R HHC are comparable for both cannabinoid receptors, while the binding of 9S HHC is tenfold lower than that of delta-9 THC. An early study using hepatic microsomes from several animals identified metabolites with hydroxylation at carbon 11, on the ring at carbon 8, and along the tail. We set out to make several of these presumptive metabolites, as well as the carboxy HHCs, to support the identification of the metabolites of HHC. This has been a success, as reports of HHC metabolism utilizing our standards continue to be published regularly. I will summarize metabolism findings on following slides, but bear in mind that new work is being released regularly, and additional data is likely to surface. And the current literature supports and expands on the identifications I will describe in upcoming slides. We will discuss metabolites identified in saliva, urine, and blood samples, starting here with saliva. This pharmaceuticals paper from 2024 identified no metabolites in saliva samples. Only the parent compounds were present. The HHCs were administered at a 1-to-1 ratio by inhalation, and yet the 9R HHC was identified at a higher concentration throughout the study. The clearance of 9S HHC looks to be 2 times that of 9R HHC based on this data. Additional literature has also been published recently in support of this conclusion that no metabolites are present in saliva samples, regardless of the route of administration. In this drug testing and analysis paper, a large panel of metabolites were identified in urine samples. While the general metabolism follows familiar THC trends, as earlier described by John, The urine samples were observed to have higher ratios of the hydroxylated metabolites over the carboxylated metabolites. Additionally, the 9S diastereomer was slightly more abundant than the 9R diastereomer. In these two papers from 2023 and 2024, both hydroxy and carboxy metabolites were identified in blood samples. Contrary to the urine metabolite findings, carboxy HHC metabolites were more prevalent than the hydroxy in these blood and serum samples. Additionally, the 9R diastereomer was twice as abundant as the 9S in these same samples. Another important aspect of metabolism to consider is route of consumption. The route of consumption can result in differing rates of clearance, and as you can see from these figures, the concentration of the ingested material over time is more varied after oral consumption, as you can see in the graphs at the top, as opposed to inhalative consumption, as you can see in the graphs on the bottom. The data in the lower graph depicting inhalative consumption also suggests earlier, overall higher peak concentration than oral consumption, and faster clearance, particularly of the parent compounds. The route of administration is just another piece of the metabolism puzzle we continue to unravel. We've just discussed the metabolism of HHC. Finally, we will discuss a compound that exists at the intersection of these categories we've established. As we continue to research metabolism and provide reference standards for metabolite identification, it is important for us to take these multifaceted compounds into account as well. Hexahydrocannabiforol, or HHCP, falls into both the analog and homolog category. Two very recent papers discuss the metabolism of the HHCP compounds. The 2024 paper described preliminary metabolism data, while the 2025 paper describes an analysis of the HHCP metabolites in urine. The 2024 paper described both hydroxylated and carboxylated metabolites, although they presumed the hydroxylated metabolites would be more significant. The primary metabolites identified in the 2025 paper in the urine sample were hydroxylated HHCP compounds, as was seen for HHC in earlier literature. The authors describe both mono and dihydroxylated HHCP metabolites, but no carboxy metabolites were present. While metabolism of these compounds is beginning to follow a pattern that will ease future identification of metabolites, there are still a few unknowns. Lastly, we will discuss our final analog of interest, the acetate compounds. The acetate compounds are metabolized quickly by human liver microsome to provide the parent compound. In this example, delta-9-THC acetate is hydrolyzed to delta-9-THC. The delta-9-THC can then be metabolized as previously described by John. The current literature consensus for acetate analogs is that after hydrolysis by human liver microsome, metabolism is identical to that of the parent. In this way, the acetate analogs are functionally prodrugs, and usage of the acetate versus usage of the parent is currently not differentiable. We've discussed the complexity of the semi-synthetic cannabinoid class and how the metabolism of many of these semi-synthetic cannabinoids resembles that of THC, both in the case study and throughout the literature. New semi-synthetic cannabinoids continue to emerge, and the class is ever expanding. We will continue to provide reference standards for identification of semi-synthetic compounds and their metabolites as the class continues to expand. Thank you so much for joining us today to discuss the semi-synthetic cannabinoid landscape, and thank you to our coworkers at Cayman Chemical who have participated in and supported this work. Jeffrey?
All right, thank you, Alicyn and Jonathan. This is an excellent overview of this emerging field of semi-synthetic cannabinoids and their metabolism. I invite any questions to be entered into the chat at the bottom of the screen, and we can go over those. One question that came in is if there was a PDF version of this presentation to be distributed, and I'll make sure a PDF version is uploaded to the workshop site discussion board. Additionally, we did have one question. So has anyone else seen interference with THC-D3? They're seeing some interfering cases with higher concentrations of delta-8 and delta-9 THC. We have not had any reports of interferences with the isotopically labeled D3. We do have a THC D9 isotopic standard as well, so that could be checked as a comparison if you wanted to have another reference standard to look at. I know there's also a question about isomerization in the metabolism work that you did, John. Was there any evidence of isomerization of delta-10 THC during the metabolism study to the 6A10A THC, or back and forth at all?
It's hard to say. When we were initially going over the results, we thought that could be a possibility. But as we looked into it further, it was kind of in the back of our minds, but I don't think we have had exactly clear evidence of isomerization. But it certainly would be something to look into as we do this work further in the future, especially if we can get better chromatographic resolution and have better, although we had really good mass spec results, but if they can be even cleaner, that'd be really helpful in answering that question.
And then Alicyn, you reported on some of the metabolites for HHC and HHCP. Were there any reports of differing rates of metabolism for those homologs?
Not that I recall seeing. It seemed like the trends were very similar.
Okay. All right, well, that was an excellent discussion. If there are additional questions, I ask that you put them in the discussion board on the CFSRE website for today's workshop. Additionally, if you need any other reference standards for semi-synthetic cannabinoids or other NPS standards, I invite you to reach out to our sales or technical services team and to search our catalog. Thank you for attending, and we appreciate all your we appreciate you all. Thank you.
Thank you for attending.